The overall goal of this procedure is to more easily visualize fluorescent neurons within thick brain slices. This is accomplished by first embedding the brain in an Agarose plug holder. The second step of the procedure is to section the brain into thick slices on the Compresstome. The third step of the procedure is to optically clear the brain slices in a glycerol gradient. The final step of the procedure is to mount the cleared brain slices onto slides for fluorescent imaging. Ultimately, results can be obtained that show high-resolution fluorescent protein reporter expression in neurons within thick brain slices through epifluorescent or confocal microscopy.
The main advantage of this technique over existing methods like thin-section serial fluorescent microscopy or multiphoton imaging is that it is much more rapid, less labor-intensive, and does not require expensive or sophisticated microscopy technique.
To begin, retrieve the mouse brain tissue that has undergone in situ fixation by cardiac perfusion and has been post-fixed for 1 to 2 hours at 4 degrees Celsius. Next, make a 2% high-strength low-gel point agarose solution in PBS and heat in the microwave until dissolved. Transfer the molten agarose to a test tube and place in a 40 degrees Celsius heat block to equilibrate the temperature. While the agarose equilibrates, glue the fixed brain tissue onto the plunger of the specimen syringe with cyanoacrylate glue. Submerge the mounted tissue in a saturated sucrose PBS solution to coat the brain surface, and then draw down the plunger into the plunger housing. Pipette the warm agarose into the plunger, completely covering the brain tissue while making sure not to introduce any air bubbles. Finally, clamp the specimen syringe with a cold chilling block for about 1 minute to set the agarose. Assemble the specimen syringe containing the embedded brain tissue into the Compresstome. Align the edge of the blade closely with the outlet of the specimen syringe and then fill the buffer tank with PBS. Rotate the micrometer 1 slice thickness forward to extrude the embedded brain tissue out of the specimen syringe. To generate the first slice, press the Start button on the control unit. Repeat the process of advancing the embedded tissue and pressing the Start button to generate the desired number of slices. Collect the slices with a transfer pipette and transfer them to a glass scintillation vial filled with PBS. Pipette off most of the PBS from the collected slices and replace with a 10% glycerol and PBS solution. Rock the vial gently at 4 degrees Celsius on a tabletop rotating shaker until the slices sink. Continue the gradual clearing by repeating the decanting and refilling process with 25, 50, and then 75% glycerol solutions. Pipette off the 75% glycerol solution and replace with 90% glycerol, being careful not to introduce any bubbles, and equilibrate at 4 degrees Celsius with gentle rocking. Now that the slices have been maximally cleared, they will no longer sink, but will instead show neutral buoyancy. Cut double-sided adhesive into an open rectangle with a 2 to 3 mm border to fit the viewable area of a 25 by 75 mm microscope slide. Using a pair of forceps, arrange the brain slices within the border on the slide. Gently lower the cover glass onto the adhesive tape, being careful not to introduce air bubbles, and apply pressure to create a seal. Aspirate any excess glycerol and then seal the edges of the cover slip by applying clear nail polish. Once the seal has dried, the slide can be stored in a slide box at 4 degrees Celsius or imaged immediately. Using epifluorescent microscopy, the expression pattern of a viral vector expressing enhanced GFP can be seen in a 200 micrometer thick coronal section of mouse brain tissue. Here, the highlighted area of the epifluorescent image is shown at high resolution using confocal microscopy to reveal individual layer 5 cortical neurons.
After watching this video, you should have a good understanding of how to prepare fixed thick brain slices for rapid and high-resolution fluorescent imaging.